Thank you. All right, so as you all know, mental disorders are by far the most important cause of disability in 20 to 50 years old. And many of these disorders actually originate earlier in life, so during development in childhood, adolescence, long before the outbreak of severe symptoms, and definitely before we detected and diagnosed the disorders. So what we would ideally like to do at some point, that's the hope for the field, is that we can use MRI for an early detection of individuals at risk before the outbreak of these severe symptoms. Now, in an ideal world, we could then just look at the disorders, right? So, for example, study schizophrenia, look at structural changes in schizophrenia, functional changes, and then identify patterns related to this disorder and apply them to individuals in the childhood or adolescence domain. But that's not really what we can do because of the heterogeneity. So if you look at structural data, you would see that there are structural changes throughout the brain, basically. And the same thing in functional data. So there's tons of reports of networks involved throughout the brain. So there's a lot of heterogeneity that sort of predicts us to form robust classifiers that we could potentially apply in childhood. And if you think about this heterogeneity, one of the sources of it is that we have so many factors that actually affect the patterns that we see at the individual patient level. So that starts with genetic effects having impact on stressors, psychopharma medication, lifestyle changes, and comorbid disorders, all of which sort of integrate into an extremely heterogeneous pattern at the individual level. And obviously, in these different factors affecting the brain, the timing is something that matters a lot. So it's obvious that, for example, effects like medication can only appear after the treatment starts, and some of the lifestyle changes and comorbid disorders are also likely to kick in later in life, whereas genetic effects are likely to have a strong impact early on. And so then again, if you study healthy adults with the disorder, you will then find, for example, strong effects of apparent brain aging, as we showed in this preprint, that we see across a range of neurological and psychiatric disorders, and this again makes it really difficult to find markers that we can use in development. So our idea for the study that I'm presenting today was to look at the genetic patterns and to map the genetic patterns into the brain. So we started out with this amazing resource, the UK Biobank, that has only now made possible this study that I'm showing to you. So here we have used the T1 data from 26,521 Caucasian individuals. And the first thing we do is we downsample the maps to FS average 4 space, just to reduce the number of vertices, because the analysis that we are running are quite computationally heavy, and so we want to reduce the high demands a bit. So we are ending up with approximately 2,500 vertices per hemisphere. And then for each of these vertices, we're running a genome-wide association analysis that basically runs a correlation of cortical thickness or cortical surface area in this given vertex with a single nucleotide polymorphisms of a given individual. So we're running a total of 10,000 GWASs here on our cluster computers. In addition, there is a lot of studies, genomic studies, running GWAS for different disorders, and fortunately they have made their genome-wide summary statistics available that we can just download them. So for example, the famous one for schizophrenia, identifying 108 loci, or several others for different other disorders and traits. And so here, specifically, we have downloaded the GWAS summary statistics for schizophrenia, bipolar disorder, major depression, ADHD, for autism, anxiety disorder, and then 2 cognitive traits. We have educational attainment and intelligence. Now, using these summary statistics, we can now run genetic correlations between the summary statistics that we have obtained on the vertex level with these from the disorders and traits. So at every vertex, for cortical thickness and for surface area, we're running a genetic correlation with these disorders and with cognitive traits. And then we're ending up with these nice genetic correlation maps, where each map now reflects the genetic correlation with a given phenotype. So in the upper 2 rows, you would see the cortical thickness, and in the lower two rows you will see the surface area. So for example, if you look at the surface area, so you would see, is there a pointer? You would see positive correlations with intelligence and educational attainment. So basically, if you have a larger cortical surface, then you're also genetically linked, more intelligent and higher educationally attained. Or for example, in schizophrenia, we would see a reduction in cortical thickness in the frontal lobe associated with a higher risk for schizophrenia. And I should say that these maps so far, they have a pretty high standard error, so they are noisy, and that is in parts because the sample size that we are using here with 26,000 individuals is still at a lower end, of course, for such an analysis. But so this is more a proof of principle that we hope to use in the future. But already with these maps, we can do something quite nice. So again back to our rationale for this study, our idea now was to ask if we can actually use these genetic maps as some sort of genetic priors to identify patterns in the developing human brain. So to this end, we made use of this extremely nice resource, the Philadelphia Neurodevelopmental Cohort, the PNC. And here we processed the structural data for 1,467 individuals. And what we're interested in the data of these individuals is not really the cortical morphology directly, but rather their normative cortical morphology. So that is, how is an individual placed relative to the developing norm. So we don't want to study age effects here. We want to know more like a crow chart, where they are put in relative to their healthy peers. So first of all, we are modeling age effects using GAM modeling and removing any associations with age at the vertex level. And we are ending up with one normative map per individual, one for surface area and one for cortical thickness. And then we are running correlations with the genetics maps that we produced in UK Biobank. So now looking at whether the individual brain morphologies or the normative maps from the children is associated with the genetic maps. And here we have developed a technique that we refer to as corticogenetic fingerprinting, which is basically we're running one correlation between each GALT normative map and each of the genetic maps. And this will result in correlations that we use as features in machine learning. And first of all, we have assessed if we can use these features to predict cognition and psychopathology in these children. So I should say that none of these children has a diagnosed disorder, so this is why we need to go into the cognitive and symptom data for like preclinical symptoms and compile summary scores. So we're doing that using PCA. So you can see that in the panel B, we're running a PCA for cognitive tests, and here I show the association between the first factor of this PCA and age, and in the panel C, it's the same thing for psychopathology. And then first thing we do is we remove this age effect, because again, we want to study normative changes, we don't want to predict age. And then we're trying to predict the normative cognitive factor and the normative psychopathology factor just using these corticogenetic fingerprints. And we're running cross-validation, we're running permutation testing, and as you can see in panel D again here, you would see that there are really nice predictions. So I think the correlation between true and predicted is around 0.27, so this is highly significant. So just based on the similarity to genetic fingerprints, we can actually predict the cognition of the individual significantly. So it's much worse for psychopathology. Still, this is significant, but here the correlation is at 0.11 or something. So this is very noisy and not quite any useful yet. But again, we are able to do that to some degree. So then finally, we wanted to assess these patterns a bit further, and we ran some linear models on these corticogenetic fingerprints, now looking into specific associations with other cognitive variables and with other psychopathology variables. So first, the left plot shows you the models for cognitive data. And here, again, you would see strong associations between the genetic fingerprints of intelligence and educational attainment with actual cognition, so that's the GF in this row, but also positive associations with schizophrenia and bipolar disorder. But what's quite interesting is that if we add in parental education, you see the same patterns here again. So we have strong associations with parental education there as well. For psychopathology, we again see very low correlations, so no strong patterns directly with the P factor. But what again quite interesting, when we add in trauma, then we see a positive genetic correlation between the anxiety map and those individuals that have experienced trauma. So this is something that we will certainly again need to replicate in data. So this is more of a preliminary finding just to show that it is feasible to run these analyses. So with that, that was a brief recap. If you're interested in this, you can either check out the bioRxives paper on it, or you can meet me at the poster later on. That's 459. So with that, I would like to thank you for your attention. Thanks the collaborators at Normand, the Norwegian Research Council, and the ARNet Neuron. Thanks a lot.